In this video, we're going to go through the urea cycle. So let's get started. So the urea cycle occurs in the inner mitochondrial membrane space, the outer mitochondrial membrane across that, and into the cytosol. So first up, we've got a CO2 and a H2O. The CO2 comes from the TCA cycle, and the H2O, the water, comes from the electron transport chain. So these together can become HCO3, bicarbonate. This plus NH4, which is ammonium, can become carbamoyl phosphate in the following reaction, where 2 ATPs become 2 ADPs plus a phosphate. And the enzyme used here is carbamoyl phosphate synthetase 1. Now, because this reaction is really important and is highly regulated, I've asterisked it in red, and I just want to go through something really important about this reaction. So what I want to go through is the commitment step. So I'm just going to cover some of this. So this is the commitment step. This causes the reaction to keep going. It's committed to completing the urea cycle after this stage. So here's the reaction here, the same as the one here. And N-acetylglutamate promotes this reaction to occur, as you can see by the plus sign in green. And it comes from glutamate and acetyl-CoA coming together. So note that acetyl-CoA is actually coming from pyruvate. If you know from the TCA cycle, pyruvate comes from the cytosol into the mitochondria and becomes acetyl-CoA. So you've got this glutamate plus an acetyl-CoA producing this N-acetylglutamate, so this N-acetylglutamate. And the enzyme that it uses to do this is N-acetylglutamate synthase, and this reaction is promoted by arginine, which is in green there, and we've got a green plus sign to indicate that it's promoting this reaction. And then it produces N-acetylglutamate, which, as I said before, promotes the commitment step, which is the HCO3 plus the NH4 becoming the carbamoyl phosphate. So that's how the commitment step is promoted, and it's regulated by mainly that N-acetylglutamate. But we also need to know where it comes from originally, so that's why we've got that reaction before that there. So that's the regulation of the commitment step. I would really recommend that you commit that to memory because it's quite important. If we move that out of the way though, we can continue with the rest of the urea cycle. So once we have this carbamoyl phosphate, a phosphate is cleaved off, and ornithine reacts with this carbamoyl phosphate and it becomes citrulline through the enzyme ornithine transcarbamylase. Then the citrulline can exit the outer mitochondrial membrane, so it exits the mitochondria and becomes in the cytosol, as you can see here. So this citrulline says, "See you later," to the mitochondria. So that's how I remember that one. Citrulline says, "See you later." So citrulline can become arginosuccinate with the addition of help from aspartate and the conversion of ATP to AMP plus 2 phosphates by the enzyme arginosuccinate synthetase, because we're synthesizing arginosuccinate. Then arginosuccinate can become fumarate, or well, rather fumarate is cleaved off when we want to make ultimately arginine. So when fumarate is cleaved off here, a fumarase can convert the fumarate to malate, and then malate can enter the TCA cycle. And we have to convert the fumarate to malate because fumarate cannot cross the outer mitochondrial membrane while malate can. So that's where that fumarate ends up. So we just discussed fumarate being cleaved off in this reaction, and it's cleaved off by the enzyme arginosuccinate lyase. Lyase enzymes cut chemicals apart. So we've got the arginosuccinate becoming arginine, and to remember that arginosuccinate is before arginine, or rather arginine is after arginosuccinate, I like to think that arginine is pronounced arginine, and I like to say arginine is next in line. So that's my little memory trick there. And then arginine, or arginine, as we've just established, can be converted back to ornithine by the enzyme arginase. And through this process, a H2O becomes a urea molecule, and that's the whole point of the urea cycle. We keep cycling through to produce urea, which then becomes urine that is excreted from the body so that we can remove toxic waste products such as ammonium, which contains nitrogen, from the body. And this is occurring in the liver. So ultimately, that's a urea cycle. We're removing those waste products from the body in the liver.